Dr. Datta obtained his bachelor's degree in molecular biochemistry and biophysics from Yale University, and later he did his MD PhD at Harvard. And after working as a postdoctoral fellow at Columbia University with the Nobel laureate Richard Axel, he joined Harvard Medical School in 2009. Dr. Datta's lab focuses on understanding how sensory cues, particularly odors, are detected by the nervous system, and how the brain uses information about the presence of salient sensory cues to compose complex patterns of motivated action on a moment-by-moment basis. This work involves studying genes to detect these odors, revealing the pattern of neural activity deep in the brain that encodes sensory maps of the outside world, exploring motor circuits and transform sensory codes into action, and probing the fundamental statistic structure of behavior itself. Dr. Datta, of course, has published many articles in different premier journals, and is a reviewer and editor of multiple scientific journals, and is associate member of the Broad Institute, and is principal investigator in the Italian Institute of Technology at Harvard Medical School Joint Program in the Neuroscience. Also, Dr. Datta has received the NIH New Innovator Award, the Burroughs Wellcome Career Award in the Medical Sciences, and the Alfred P. Sloan Research Fellowship, the Searle Scholars Award, and the Valley Young Investigator Award, the McKnight Endowment Fund Scholar Award, and now recently has been named Fellow for the National Academy of Science Cavill Scholars Program. So, and in addition to all these accolades, Dr. Datta is co-founder of Optogenix, no? And which manufactures biocompatible optic fibers for recording manipulation in the brain, and two other companies, Syllable Life Sciences and Aveline Therapeutics. He's joined with Dr. Taralyn Tan, who is the Director of Educational Programming at the Harvard Medical School Department of Neurobiology and the Graduate Program in Neuroscience, who will be talking to all of you about the graduate program at her institution after the talk from Dr. Datta. So thank you so much, Dr. Datta, for being here. Tari, thank you for joining us this afternoon. And I'll leave you, everyone, to listen to a wonderful talk. Thank you.
Thank you. Okay, can everyone see that?
Yes, we can.
Wonderful. So thank you so much for that incredibly kind and generous introduction. You know, this pandemic has been, I think, for all of us, you know, truly horrible in all sorts of different ways. And one of the only things that makes it bearable is the chance to, you know, get to share science. And for me, it's particularly exciting to get to share science with, you know, up and coming new scholars. And so for me, this is a real treat and a real privilege. And I thank you all for attending. Also, I would really encourage you to stick around to hear what Tari has to say at the end of my talk. She's amazing. She may or may not admit this, but she was a graduate student in my laboratory. And she's a spectacular advocate for graduate education. And really passionate about, you know, helping to build the next generation of neuroscience scientists. And so I'd really encourage you to hear what she has to say. I think, you know, we at Harvard are really interested in building up neuroscience as a field. And so, yeah, I would encourage you to seriously consider Harvard when you think about graduate school in the future. So why don't we just, let's get to it. So this is a movie of a deer mouse foraging for food in the wild. And if you just look at this movie for a while, you can, I think, quickly discern something interesting about the strategy this mouse is using to try to find food. So what this mouse is doing, and you can just see this by eye, right, is that every couple hundred milliseconds or so, this mouse moves its body. It changes the direction it's facing, and maybe takes a step forward. It maybe extends its whiskers a little bit. And in doing that, it's collecting some sensory information. And then on the basis of that sensory information, its brain decides to do another thing 100 milliseconds later. And it emits that little behavior. And it does this over and over and over again sensory leading to motor, motor leading to sensory, and on and on and on. And there's this kind of parade of micro behaviors the animal generates, which somehow coherently add up to this animal seeking food. And we know that in the wild, animals often have to put together these little behaviors into the appropriate sequences. And there could be hundreds or thousands of these little behaviors that are placed in just the right order in order to let animals like this succeed at the task that they have, which in this case is to find food. But other tasks that animals face in the wild are things like avoiding predators or finding a mate or finding safe harbor in the rain. And so in truth, this is kind of the broad set of questions my laboratory is interested in, right? We're interested in understanding how the brain organizes information about the sensory world. And that's information that can vary on time scales of, say, 10 milliseconds or so, the fast time scales. And your brain takes advantage of this sensory information to compose ongoing patterns of action. Every 100 milliseconds or so, as you saw in the movie I just showed you, the animal chooses what to do next. And it makes its choices based upon the things that it's sensing. And then through the actions it takes, the steps it took forward, the turn to the right, the step to the left, lifting the snout up to sniff, through the actions, it changes its view of its sensory world, right? And it gleans new information about what's around it. And the cycle goes on until the animal solves its problem of existence. And so my lab is interested both in how the brain organizes sensory information in order to facilitate action, and it's interested in how different actions let the animal understand more about the world around it. Now, we want to understand ultimately how the brain supports both of these processes. And to a first approximation, solving this problem of sensory-motor coupling is what the brain evolved to do. All animals, humans, mice, worms, fish, all animals have to solve this basic set of problems. And so because the brain evolved to solve this problem, we thought that maybe a useful way of addressing this set of challenges would be to think about the sensory modality that is most important to most animals in the wild when they solve their problems of existence. And that, of course, is the sense of smell. Most animals rely deeply on their sense of smell to interact with the world. And so my lab focuses on trying to understand how smells from food, predators, or mates enables the brain to drive different patterns of action, and then ultimately how those different patterns of action let the animal make better predictions about the olfactory world around it. So as was mentioned, my lab was started about 10 years ago, and we had a pretty simple seeming goal at the beginning, which was to try to understand how sense changed the behavior of mice in the lab. And then we realized quickly as we began to confront this problem that the only way to really understand how a stimulus changes a behavior is to be able to measure behavior. And the behaviors that we cared about were naturalistic, spontaneous, untrained behaviors that are freely expressed by mice, either in the lab or in the wild, when mice aren't restrained or overtrained or even rewarded. And in thinking about how to measure behavior under those circumstances, we quickly realized there was a kind of technical gap that was preventing us from understanding from a quantitative perspective and a comprehensive perspective, what the actual structure of behavior is, so that we can understand how the brain might change it when, for example, the brain encounters a smell. And to give you a sense of exactly what I mean, let's take the very simplest example possible of a mouse behavior. So here we are looking with just a regular video camera down at a C57 Black 6 mouse that I placed in an apparatus called a circular open field. And I'm sure all of you know this is really just a bucket that I bought at the Home Depot, right? So let's watch this animal as it behaves. And you'll notice something, even if you've never studied mouse behavior before, about what's going on here. So I put this mouse in this strange environment, and the mouse doesn't freeze up, and he doesn't just do random stuff. Instead, the mouse's brain recognizes that the mouse is in a new place, a new environment, and in response to that, the mouse's brain emits a series of coherent motor commands that encode a pattern of behavior that's predictable, that we would call as humans locomotory exploration. We're watching this animal explore this environment to try to understand what's in it, right? And so those are not random behaviors, they're very structured behaviors. And the mouse's brain is creating a kind of time-varying structure of behavior that allows this animal to sample information in this new little universe I've placed him in. And the question is, how do we quantify what and describe what this actual pattern of behavior is and how it varies over time? And with the tools that have traditionally been used and that most people use, the things that you can measure that this animal does are things like, how fast does the animal go, or where in the bucket is the animal, or how close to the wall is it, or how often does it enter the center of the arena. And all of these things are important aspects of the animal's behavior, but none of them capture the flavor of what we're talking about. Right? What we really want to know is, what are the parts of behavior out of which this animal composes its overall pattern of action, right? How do those parts vary over time? The idea that this animal's behavior is richer than just its velocity or its position, that's not my idea. It actually comes from work that a bunch of scientists called ethologists did 50 or 60 years ago. And the ethologists believed that this animal's behavior, like the behavior of all animals, is actually composed from a series of stereotyped, brief, reused modules, which in the case of this mouse are going to be like the first half of a rear or a turn to the right or a step to the left, out of which the brain can build a variety of behavioral sequences that encode any behavior you can think of, including this locomotory exploration. So essentially, to test this ethological hypothesis that behavior is built out of modules, over the past several years, my lab has been working to build machines that can identify automatically from behavioral data any underlying modules out of which behavior is organized or created. And the idea is that if I can somehow identify and count up all of the modules of behavior that this mouse is emitting, I can use that information to create a much richer description of behavior than just telling you, where in the arena the mouse is or how fast it's moving. And also, I should be able to do things like make predictions. I should be able to predict a mouse's future behavior based upon its present behavioral state. And most importantly for my lab, if this project works, I should be able to make predictions about what's going on in the animal's brain while the animal is generating various forms of behavior. And so today I'm going to tell you a little bit about a technology that we've built that helps identify modules out of which mouse behavior is built. This technology is called motion sequencing, or MoSeq for short. And we actually described it in some detail in a paper that was published about 5 years ago. And so because of that, I'm not going to bore you with any of the technical details about how, how MoSeq actually works. So during the question and answer period, I'm happy to talk all about it. Instead, I want to give all of you a very high-level overview, just conceptually, how does MoSeq go about identifying the modules out of which behavior is built? So MoSeq really has two parts. It has a kind of data acquisition component where we measure something about an animal's behavior. And I'll tell you right now, we can measure lots of things about animals, different animals' behavior, as we'll show you. And MoSeq can process all sorts of different kinds of data to try to identify underlying modularity that reflects the structure that's inherent in behavior. In my lab, we tend to use a single type of data because it's really, really informative. And that is, we tend to look at 3D movies of mice as they behave in any arena we choose. So if you look at the upper left up here, what you're seeing is a 3D movie of a single mouse exploring one of those circular open fields, one of those little buckets. And so the way these 3D cameras work is we place a camera from a single point of view, like we just basically replaced the normal camera that I showed you in the previous movie. We just replaced that with one of these depth cameras. And these depth cameras are capable of seeing in 3D. And so you see this mouse has a heat map over it. It's kind of heat mapped. That heat mapping reflects the mouse's height at any given pixel on the mouse. So when the mouse's head is white, it means the mouse is kind of pointed upwards and kind of sniffing up towards the camera. When the mouse's back is white, it means the mouse's back is arched upwards towards the camera. And you can get a sense for that just by looking at this movie. Okay, so we have this 3D data that basically captures rich information about this mouse's three-dimensional pose dynamics. We then feed those data to the MoSeq algorithm, which is really a model for behavior that is based upon the ethological hypothesis that behavior is built out of parts, it's built out of modules. I won't explain to you the details of how this model actually works, but I will tell you that it works in an unsupervised fashion. And what that means is that that MoSeq identifies behavioral modules from a mouse experiment, like this mouse experiment I'm showing you here on the upper left, without us as humans having to say anything about what we think behavior is. So I, as an experimenter, don't tell MoSeq, oh, go find me some running, go find me some rearing, go find me some sniffing. Instead, whatever behaviors the animal generates in an experiment, MoSeq automatically identifies those behaviors because they're repeated patterns of action and labels them as behavioral modules. And so really the output of MoSeq is kind of like a parts list of behavior. It's a list of all the different modules that an animal expresses within a given experiment and the order in which they occur. And because we believe each one of these modules is a key component of mouse body language, we have chosen to refer to each one of these little modules as a behavioral syllable. And we talk about the ordering between these syllables over time as animals express different syllables as behavioral grammar. So let me show you what one of these behavioral syllables actually looks like to give you a sense of whether these behavioral syllables are really a kind of parts list for mouse behavior. So on the left is again a 3D movie of a mouse and in one of these little circular open fields. And when I hit play, the mouse is going to generate some actions. At some point, a dot is going to come over the animal. When the dot comes over the animal is going to express a little motif of action that has been identified by motion sequencing as an individual behavioral syllable. Now, I've seen this syllable many times, and so I have decided after the fact, post hoc, to give it a name. And I'm going to call this syllable the move it along syllable. Okay, so when I hit play, you'll see the animal generate some behavior. There's the dot, and the animal kind of scooches forward during the expression of the move it along syllable. Here you can see the movie looping just to give you a flavor for what the syllable looks like. So is move it along really a component of behavior? Is it a stereotyped motif out of which more complex behavior is built? On the right here are 30 separate examples of this same behavioral syllable, except they're drawn from 8 separate experiments and actually 8 separate mice. So really there's these kind of 30 completely different examples, right? You can think of them as completely different. And for convenience, I've decided to overlay all of these individual examples into a single master movie and time lock them all together, okay? And so because they're all different examples, when I hit play, you'll see these mice generate all sorts of behavior. But when the dot comes over the animal, coherently, these mice all generate this move it along syllable. As you watch this movie loop, you'll notice that there's a certain amount of spatiotemporal variation associated with the implementation of this particular syllable. In other words, sometimes the syllable is short in some instances, other times the syllable lasts longer. And if you look at the shapes of the body that the mice are taking as they express these syllables, it's not always exactly the same. Sometimes they're scooching around a little bit more, sometimes they're scooching around a little bit less. And that is entirely by design. MoSeq is designed to identify stereotyped units of behavior to recognize that each time an animal expresses a behavior, it might be slightly different, okay? So and we refer to these movies on the right as crowd movies, and they're useful because they give you a sense of kind of what each behavioral syllable is. Here are 6 more of these crowd movies that show you 6 different behavioral syllables that are identified when mice are exploring the circular open field. Some of these are very obvious. You can see this rear up syllable, it's just the animal rearing up. Some of these are pretty funny. The one on the lower right, this get out syllable. After mice have been in a bucket for like 10, 15 minutes, they get super bored. So they want to escape. And you can see the mice kind of migrating themselves to the side of the bucket and then kind of jumping out as they try to escape the bucket. So these are 6 different examples of the behavioral syllables that are identified automatically by MoSeq, typically depending on the data set that we feed to MoSeq. In the open field, if you just have wild type mice, behavior is made up of something like 40, 50, 60 different behavioral syllables. And each one of these is a sub-second motif of action. The average duration of each one of these little syllables is about 300 milliseconds long, okay? So what you're looking at here is MoSeq identifying for us some of the parts of behavior. How can we think about these parts being placed into sequences over time to compose, real, actual, naturalistic action like the locomotory exploration I showed you a few slides ago? So we can just take advantage of the kinds of tools that have been handed to us by folks who think about like bioinformatics. So here in the lower left is a behavioral state map. Each little node in the state map represents an individual behavioral syllable, and the diameter of each one of these nodes reflects how often the syllable is used. So if a syllable is only used occasionally in an experiment as a small dot, if a syllable is used a lot in the experiment, there's a big circle. you see that these circles are interconnected by yellow directional arrows. The thickness of these arrows reflects the observed likelihood of transition between one syllable and the next. In other words, these yellow lines tell you the rules that govern the ordering of different syllables, right? So if two syllables aren't connected by a yellow arrow, they don't follow each other in time. But if they are interconnected with a yellow arrow, it means that they tend to follow one after the other as animals express behavioral sequences. So these behavioral state maps let us do something really rather remarkable, right? They let us tell the future. Because I know that if a mouse is expressing, say, this behavioral syllable over here, that in the next moment, it's likely to be expressing this syllable, and then this syllable, and then this syllable, and so on and so forth. And so we can do one of the things that I kind of laid out as one of our goals, which is to if we're appropriately analyzing behavior, it lets us make predictions about a mouse's future behavior based upon the mouse's present behavioral state. The other thing, obviously, that this kind of behavioral state map does is it objectively, comprehensively, and quantitatively summarizes every action that a mouse takes within a given experiment, at least from the data that we're recording, in this case, the data that come from a 3D camera. And that in turn means that we can use these kind of behavioral state maps, these summaries, to make comparisons between mice as we manipulate them in various ways. And so my lab, in collaboration with other labs, has done all sorts of experimental manipulation to mice. We've changed the genome of mice. We've given mice drugs. We've changed the environment the mice are in. We've directly activated neural circuits. And in all cases, we see from these motion sequencing maps, exactly the behavioral changes that we would expect to see by human observation alone. But we also nearly always learn something new. There's some features of behavior that we as humans just aren't good at recognizing, but motion sequencing captures the syllables that have changed and the sequences that have changed in a way that's really organized and is much better than human intuition. And that has offered us a quantitative lens, really, to understand how all sorts of experimental interventions change mouse behavior. And that in turn, in the long run, is going to help us link genes to neural circuits in the brain, to behavior, to the output of the brain. And so we think this is a very powerful tool, ultimately, for understanding brain function. Now, there's something that I'm kind of implying here that I'm not really giving you any proof for, so you should be skeptical, okay? So I'm implying, but not proving, that this newfangled way of thinking about behavior as being composed out of parts is actually better than what we've been doing forever, which is to kind of track the position of the mouse or its direction or its speed. Is that really true? So Alice Wilcho in my lab did an experiment, which we just published this week in the journal Nature Neuroscience, so I would encourage you to go look at it if you're interested in the latest, to try to test the idea, which is really a hypothesis, that thinking about behavior as being built out of parts will provide us with a better description for behavior than just using the traditional ways of thinking about behavior. And so here's what he did. He injected nearly 1,000 mice with one of about 20 different neuro or psychoactive drugs at one of 2 to 6 doses. And as you know, when you give a neuropsychoactive drug to a mouse, those drugs change the mouse's behavior because they're activating different neural circuits or inhibiting different neural circuits. So he took all these drugs and all these doses, and he injected a ton of mice with all these drugs and doses. And then he just threw these mice into the bucket and filmed each mouse for 20 minutes, okay? And then for each mouse, he built 2 different kinds of behavioral fingerprints, basically. The first kind of behavioral fingerprint is the traditional behavioral fingerprint. How fast was the mouse moving when it was on this drug? Where was it in the bucket? So on and so forth. And I'll show you the things we measured on the next slide. So kind of traditional fingerprint. The other kind of fingerprint was based on MoSeq. We just measured, what are all the different behavioral syllables and how often they're used. So given these 2 different behavioral fingerprints, or these 2 different ways of describing the behavior of the same mouse, we could then feed those data to something called a linear classifier. And the linear classifier's job, it's a computational tool, and its job is to make predictions. And we asked the classifier, can you, based upon these 2 different types of data, predict accurately which drug and dose each mouse was on? And we would decide, which description of behavior was more informative based upon the ability of this linear classifier to make predictions. And I'll show you all that over the next 2 slides. So here's just some of the raw data to give you a flavor of the actual experiment. On the y-axis are some of the drugs we tested at some of the doses, a subset of the doses. And here in the middle, where it says scalars, those are a small subset of the kinds of traditional things we measured about behavior. So we fed our classifier the complete distribution of all the positions the mouse adopted in the arena, the complete distribution of all the speeds that the mouse took, the distribution of the lengths of the mouse's body. We actually, just to give ourselves a disadvantage, included information of the complete distribution of the height of the mouse, which is actually something you can only really get with a 3D camera, but we threw it into the traditional measures anyway, just to make our job tough. And what's interesting is that if you just look at these raw data, you instantly understand why for 100 years people have been characterizing animal behavior by measuring speed or position, right? The reason why is because it gives you an immediate intuition for what's going on in some cases. So if you look at haloperidol, right, that's a drug that we know in humans knocks people out. And if you look at high-dose haloperidol right here, up here, you'll see that the position of the mouse is kind of fixed in the arena and the mouse's speed is 0, right? And that's because the mouse has been knocked out by the drug, and you can just tell that in this graph. Conversely, if you look at methylphenidate, right, that's a strong stimulant, it's a strong stimulant in humans. You see that the position of the mouse is at the edge of the arena and the speed is basically infinitely fast. And that's because methylphenidate is acting as a stimulant, and you can just see that reflected in the mouse's behavior as it circles really fast around the edge of the arena, which is just in the data. You can see it with your eyes. For MoSeq, we just made a little barcode that reflected how often each behavioral syllable is used. And compared to these traditional data, MoSeq, these barcodes look useless, right? They don't really tell you anything about the behavior the animal is generating, right? I mean, it's just some nonsensical barcode. But we took these two different types of data and we fed them to a linear classifier. We learned something very different. So what you're looking at here is a kind of plot called a confusion matrix, okay? On the y-axis are all the different drugs we gave to the mice, listed out. On the x-axis are all the drugs that the linear classifier thought the mouse received based upon behavior alone. And so if the linear classifier is doing a perfect job at predicting which drug the animal actually got and is making all the correct predictions, you'd expect this confusion matrix to have a bright white line on the diagonal and black off the diagonal. That means that the classifier is capable of making perfect predictions based upon behavior about what drug the animal got. And if you just look by eye at these graphs, what you learn quickly is that traditional ways of measuring behavior aren't great at telling drugs apart. They're basically useless. But 97% of the time, with data based on MoSeq, I can predict exactly which drug an animal is on. And 93% of the time, I can predict both the name of the drug and the specific dose the mouse received. And this quantitatively proves that thinking about and describing behavior is being built out of syllables, out of these parts, and out of sequences, you know, how these parts flow over time. That way of describing behavior, which was, of course, first discovered and invented and conceptualized by the ethologists 60 years ago, provides you with a much more powerful description of behavior than just kind of agglomerating the traditional ways of the traditional metrics that we obtain when we track a mouse's position or track its speed or track its height, for example. And so for us, this was really gratifying because it suggested to us that this machine that we built, this motion sequencing machine that we built, was really, from a quantitative perspective, capturing important and subtle features about a mouse's overall behavioral state. Okay, so let me show you an example. Let me just check the time. I want to make sure I don't take too much of your time. Okay, I have a little bit more time. Let me just show you a couple examples of how we've been using motion sequencing to give you a flavor for sorts of things you can learn and what this car can do. So I want to show you an example that's drawn really, of course, from my favorite subfield, right? I'm really interested in how smells change behavior. And so this is one of the first experiments we did, and it turned out to actually be one of the most important we've ever done. Here you're looking at a bunch of mice that are being thrown into a box. And again, I'm going to overlay 30 separate experiments. And so what you're actually seeing are the behavior of 30 separate mice in this box, all overlaid. Instead of showing you a 3D movie here, I'm just going to outline each mouse's body with a little squiggle. And I'm going to average the position of all the mice which they took throughout the experiment in gray in the background, okay? And at the beginning, and the last thing I'll say is that each of these corners contains a little port and I can blow odors through each one of these little ports on all the corners, okay? So if when I hit play on this movie, what you're gonna see is what happens if there's just air in this box. And so what you're looking at is basically normal exploratory behavior of mice when they're thrown in a box, right? They dart through the center because they don't like open fields. They love exploring the corners and hanging out in the corners, right? Normal behavior. Now look at what you see if in the lower left, I blow the odor of a fox. So that fox odor scares these mice, right? So they think they're gonna get eaten in this box. So they kind of sniff the odor and they run away and they hide in the upper right corner away from this putative fox, which is gonna eat them in the lower left corner, right? And so it's clear the odor has changed the overall pattern of the animal's behavior. And the question is, from the perspective of MoSeq, what happened? We know that new behaviors are being emitted as a consequence of exposing the animal to an odor. What does MoSeq say actually happened and changed in the animal's behavior as a consequence of being exposed to the scent of a fox? So here are two state maps, before and after TMT, which is the name of the chemical odor that we use that comes from fox. And if you actually go back and forth with your eye between these state maps, what you'll quickly learn is that they're basically the same behavioral syllables under both conditions. Okay, so all the different motifs of behavior that are expressed in the control condition, those same little syllables are all present in the TMT case. So there are actually no new behavioral syllables, modules, motifs that are being expressed, despite the fact that we know that TMT is eliciting a huge number of new behaviors, right? Escaping the odorant, freezing in the corner, all those things. So where do those behaviors come from? Well, as you can see from the summary here, what has really changed are the interconnections over time between behavioral syllables. What's changed isn't the parts list for behavior, but the sequence in which they tend to occur. And if we subtract off these two behavioral state maps, what you can see here in red are all the behavioral sequences that have been downregulated, and in blue are all the interconnections between behavioral syllables that have been upregulated as a consequence of being exposed to this TMT odor, okay? And so every new thing the mouse did in this experiment, all of the new behaviors the mouse generated, basically are trajectories through the blue part of this state space. So let's look at some behavioral sequences. So now when the dot comes over the animal, you're not looking at a single syllable, you're actually looking at a series of 3 blue syllables in a row. And let's ask what they are. And you can see here on the left, these blue sequences encode the freezing behavior, which we know is a thing that happens when animals are scared and certainly happens in response to this fox odor TMT. We also see the motivated avoidance behavior here in the right movie, right? You can see that the animals run away from the odor source, and that's another sequence that is upregulated as a consequence of being exposed to the fox odor. Okay, so conceptually, what does this tell us? So I'd argue it tells us something pretty profound, right? It tells us that when animals are exploring their environment naturally, their behavior is built out of a set of parts, and the ordering of those parts is flexible and adapts to the environment around the animal, such that when an animal is, for example, confronted with a predator, it doesn't just generate completely new behaviors, but instead generates adaptive actions by resequencing previously existing behavioral syllables. So if that's true, there's got to be somewhere in the brain that's basically in charge of placing these syllables in the correct order, and is in charge of adapting that order to new information that's coming into the brain from the outside world. So where might that be? So my lab has been thinking about this very seriously now for the past several years. All the work I'm going to show you today related to this is work that was done by a postdoctoral fellow of mine named Jeff Markowitz, who's a joint postdoc with my friend and collaborator Bernardo Sabatini, along with 2 graduate students in my lab, Wynn Gillis and Maya J. So we've been thinking about this problem, where in the brain my behavioral sequences be assembled, and that naturally, after doing a lot of reading and thinking and talking to people, led us to think about a structure in the brain called the basal ganglia. Now, for the sake of this talk, I'm not going to give you a long lecture about the basal ganglia or all the different nuclei and how they're interconnected and who talks to whom. I'm sure in your basic neurobiology classes, many of you have seen a diagram like this before. I'll just remind you that the input nucleus to the basal ganglia is this part of the brain called the striatum, and decades of work in humans and in animal models have led to a kind of consensus, which is that the striatum plays a critical role in implementing behaviors. So as we move our bodies in real time, the striatum helps control action initiation, action invigoration. It helps us execute behaviors. But also, the striatum is in charge of action selection, the process through which we choose what to do next. Furthermore, in a specific subregion of the striatum called the dorsolateral striatum, there was some evidence that suggests actually that in the context of some behaviors, the dorsolateral striatum might be important for various forms of behavioral sequencing. And so that all led to a kind of hypothesis that maybe the dorsolateral striatum actually encodes information about behavioral syllables or behavioral grammar. Right? And if we could find a neural code, if we could find neurons in the brain that represent behavioral syllables or grammar, that would explain to us why it is MoSeq provides such a nice summary of behavior, because the brain actually organizes behavior out of these little syllables and through behavioral grammar. So to test that hypothesis, my laboratory has rendered motion sequencing compatible with all forms of in vivo neural recordings, okay? Those include some techniques that I'm sure you've heard of, like extracellular electrophysiology, some newer techniques like fiber photometry, and even a really exciting new technique called miniscope technology. And I'll show you that on the next slide. So this is, here you're looking at an experiment. I'll explain all of the panels in which we are simultaneously performing motion sequencing and characterizing the 3D behavior of mice, while at the same time, we are using a miniaturized microscope that I've implanted in this animal's dorsolateral striatum to measure the neural activity of individual cells, individual spiny projection neurons, which are the main neuron type in the striatum. So here in the lower left, this is just a mouse in the circular open field, and you can see just the position of the mouse as it runs around in this little bucket. On the upper left, you see a 3D movie of the mouse. And this is different from the 3D movies I've shown you before, because here we've cropped out the digital image of the mouse, and we've rotated it such that its nose is always to the right and its tail is always to the left. This is a mouse, as you can see on the bottom, that's freely running around in a bucket. Now, a thing you'll notice is that this mouse is wearing a hat, and out of this hat is coming a large cable. And that is because we've actually implanted this miniaturized microscope inside this animal's dorsolateral striatum, and we're using this cable to kind of visualize neural activity in the animal striatum. So on the far right, you're looking at a movie of this mouse's brain, and the flashes of light you're seeing are individual neurons that are firing action potentials as this mouse runs around. So we can analyze this data. So you see in the middle lower panel, what you're seeing are traces of activity, firing patterns of individual neurons during this experiment. And because we can do motion sequencing simultaneous as we measure neural activity, above it you can see the different behavioral syllables that MoSeq is identifying on a moment-by-moment basis. And if you just stare at this middle movie for a little while, what you kind of quickly appreciate is that as animals switch from one behavioral syllable to the next, there tends to be a burst of activity in the striatum that accompanies changing from one behavior to the next in time. And if you do what we did, which is to look very carefully at which neurons are activated when animals switch from one syllable to the next, what you realize is that there's a neural code for behavior in the dorsolateral striatum, where the specific neurons that are firing actually represent or tell you about what 3D syllable is being expressed in that very moment. And we can prove that in an interesting way. This is on the left side of this plot, through something called a classifier. So I want to explain this in a little bit of detail because it's fun to understand. So imagine you have a brain that only has 10 neurons in it. And 5 of these neurons fire when the animal's walking, and the other 5 fire when the animal's sleeping. Okay, imagine you had a brain that just worked that way. And imagine you had a camera and you could assess neural activity in this animal. Whenever the first 5 neurons fired, you would know that the animal was walking. And whenever the second 5 neurons fired, you would know that the animal was sleeping. Right? And that's because there's a 1-to-1 correspondence between when the neurons fire and when the animals generate a certain kind of behavior. And so we can basically quantify how closely related neural activity is to behavior by asking how well can we predict behavior, ongoing behavior, based upon activity of a set of neurons. And that's what this plot on the left actually shows. On the x-axis is neuron number. That is, if we feed our classifier 10 neurons, we have less information. If we feed it 600 neurons, of course, it's many more neurons, and so that's much more information. So we can ask how information content varies with the number of neurons. And on the y-axis is just how well can we identify which specific syllable is being expressed at any given moment in time. And I want you to think about how hard this task is, actually. So there are about 50 syllables that an animal can express during a given experiment. And so what we're asking this classifier to do is to take a neural activity from the brain, okay, and predict which of 50 things the animal is doing. That's like basically asking someone to be able to predict which card is coming out of a deck, right? Is it the Jack of Spades? Is it the Queen of Diamonds? And 25% of the time, our classifier gets this right perfectly. And if we relax the decoding criteria a little bit and say, can you identify based on neural activity whether the animal's expressing, say, a rearing syllable or a rearing syllable that's very similar to that first syllable, if you kind of group syllables a little bit, this decoding accuracy goes up to 80%. And so there's a ton of information in the striatum that encodes which three-dimensional syllable the animal is generating and expressing at any given moment in time. On the right, I'm not going to explain this plot. It's a little bit complicated, but all it shows you is that if you look at individual syllables, the actual neural activity that's associated with an individual syllable varies depending on the specific sequence in which that syllable is embedded. So if a given syllable is part of a high probability sequence, there's actually not that much activity. And if it's part of a low probability sequence, a series of syllables that's unlikely to happen in the context of the experiment, there's a little bit more activity. And what that means is that we not only can decode from neural activity what the animal is doing, what syllable that's expressing, we can also decode the sequence content, the ordering, the grammar with which that syllable is being expressed. And so the striatum encodes explicit neural correlates, both for the syllables that MoSeq expresses and for the grammar that MoSeq reveals. And the key question is, of course, which one of these things does the striatum actually control? Does the striatum control the expression of each of these syllables? Does it control the assembly of behavioral sequences? Does it control both? Does it control neither? So we did a very simple experiment. We just injected a chemical called NMDA into the dorsolateral striatum of living mice, and it turns out NMDA kills neurons. And so we destroyed this animal's bilaterally, both of its dorsolateral striata. And we can then perform motion sequencing. And so here you're looking at 3D movies of mice that lack their dorsolateral striata. And what you can see is that they look pretty normal. And if we actually do MoSeq, these animals look pretty much intact and normal. And the syllables they're expressing, they express all the same syllables, and the morphology of the syllables is correct. But if we go ahead and compute behavioral grammar, and we make a state map, we see something very different. So this is the kind of state map where all of the syllables are on the outside of the circle. And so the white lines are like those yellow lines I showed you before. They just are meant to indicate the interconnections over time between syllables. And as I showed you before, if you look at the sham case, the control, there are rules that the brain elaborates that governs the ordering in which these behavioral syllables can occur. But if you lesion the dorsolateral striatum, those rules go away. And all of a sudden, syllables are interconnected willy-nilly, seemingly randomly over time. Now, so this is a very impressive result, right? Because it says that the dorsolateral striatum is not required to implement behavioral syllables, but is required to assemble them into behavioral sequences. That being said, I just showed you a movie of a mouse with a lesion striatum, and that mouse looked pretty good, right? Looked pretty normal. So who really cares about this result in the sense that it doesn't seem to affect the animal's behavior very much? But I will remind you that TMT, that fox odor, changes behavior, and the way it changes behavior is by causing the animal to resequence its behavioral syllables. So the question is, can a mouse with a lesion dorsolateral striatum actually respond to TMT? So let's go ahead and look at a sham. The sham approaches the TMT port, smells it, and runs away because, again, it thinks it's going to get eaten by a fox. And if you wait a minute, you'll see that because mice are idiots, this mouse will reapproach the TMT port, smell it again, and run away again. If, on the other hand, we take a mouse that has a lesion, the mouse doesn't explore the TMT really at all. We know from a sensory perspective the mouse can sense the fox odor, but the mouse is incapable of modifying its pattern of behavior and reassembling these behavioral sequences in order to generate the adaptive avoidance behavior that we know TMT should elicit. And this proves that the dorsolateral striatum is responsible for assembling behavioral grammar. So now, just for reasons of time, I'm going to skip over a bunch of science. I'm going to show you one last exciting thing before I turn this over to the question and answer period. So I'm going to not show any of these data, although they're very interesting. Social mosaic, very exciting. Lots of interesting things happening there. I just want to show you this. So as I mentioned at the beginning, mosaic has two parts. It has an analytical engine that discovers structure in data. And it has a data entry kind of component where we measure behavior, and typically we feed mosaic these 3D videos. But we can feed mosaic anything. So we've been feeding it all sorts of stuff. In this case, we're feeding it 2D video. This poor subject on the left has her hand on a hot plate. And every once in a while, a hidden investigator will turn the hot plate on, and it'll cause a painful stimulus to this woman's hand, and she will grimace. And we can actually compute over these videos and identify facial syllables that capture the organized structure of her behavior. And believe it or not, we can use these facial syllables to accurately predict how much pain she's in. And so we've been using the mosaic to analyze not just mouse behavior and rat behavior, and actually the behavior of all sorts of different mouse species and strains, but also beginning to port this to looking at humans. We've used it to look at human video data. We can use it to analyze actigraphy data or accelerometer data on the kind of smartwatches that everyone wears. And so mosaic really represents a very general tool for characterizing and exploring behavior. And with that, I'll end. I'll just mention that mosaic was invented in my lab by Alex Wilchko and Matt Johnson, and much of the work that I showed you today was carried out by a suite of really spectacular researchers who I mentioned, including Jeff Markowitz, Wynn Gillis, Caleb Weinreb, Rockwell and Yoha, and Ralph Peterson, along with Maya J more recently. And this is a picture of the lab. Thank you so much for your attention, and I look forward to taking your questions.
Wonderful. Wow. I'm speechless, actually, because I'm a behavioral neuroscientist, and I just found the holy grail. Oh my God. I was like, what? And these are the events in science where you see a leap on what we have as things available, and now we have these possibilities. And I really, my appreciation to all the hard work on your lab and the things that you're doing are amazing. And I really, I tell you, I'm like, oh my God, all the possibilities that for neuroscience.
It's very kind, thank you.
No, it is. It was, everybody's so excited, and there's some questions, I guess, if you like, I can, pronounce them, or maybe Tari wants to, I don't know, it's up to you guys. I usually read the questions, but if you want to read them by yourselves, that's fine with me.
I'm not going to read them, so if someone else can read them, that'd be great.
Okay. So we have a question that it says, this is from one of our students. She's Viviana. She says, have you used mosaic to study dynamics between dominant versus subordinate mice?
I have, and those were the slides that I skipped over. So a major thing that we've been working on is to try to do mosaic on 2 mice. Right, so you can imagine that's a real challenge, and from a technical perspective, it really is, and it's a challenge because when 2 mice are behaving, they overlap with each other. And if we're looking from above, right, like with these cameras, our view of the 2 mice is kind of obscure, right? And so we had to invent mathematical techniques that allow us to deconvolute these 2 individual mice and to fill in the missing parts of their bodies. But we have succeeded at doing that. And having done that, we can now take mice that are in social interactions, and we can do mosaic. And so what we see is that if we take 2 singly housed CD1 males and we put them into a box together, on first interaction, if you just measure their behavioral syllables, they're pretty similar. And then the second time you do it, they become a little bit less similar. And the third time you do it, they become a little bit less similar. And that's because one mouse is clearly becoming dominant, and the other mouse is clearly becoming subordinate. And we're watching the emergence of dominance and subordinate behaviors basically in real time. So we've been coupling that recently, you know, I showed you that we can put these little miniaturized microscopes in the brain. I showed you data from the dorsolateral striatum. Now we're doing it in prefrontal cortex. So we have a camera in PFC as we watch these social interactions unfold. And what's interesting is that if you look at neural activity in PFC, we actually see neural representations, not just for the behavioral syllables that I was showing you that are also represented in dorsolateral striatum, but we see representations for social syllables, where one animal is doing one behavior and another animal is doing a kind of linked behavior. And so we're beginning to explore, this is very preliminary, but we're beginning to explore neural codes for social syllables, and it's going to be an important kind of venue for us going forward. We're really interested in this space.
Mm-hmm. It's very important as well. And there's another question. I was wondering if there's any relevant connections between the dorsal striatum and the barolateral amygdala that might also help to understand the changes in response to aversive order.
Yes, that is an extremely sophisticated question. I just want to say right off the bat, that's a great question. So, and it gets at a kind of interesting point, which is we are used to, those of us who have spent any time thinking about the basal ganglia, and probably those of you who've taken a survey course, are used to thinking of the dorsal part of the striatum as being involved in kind of actions, and like, you know, action selection and composing action and movement. And we're used to thinking of the ventral part of the striatum as being involved in emotions, right, in valence. And so when you give a negatively valenced odor like TMT, your expectation is that the amygdala is going to be involved and the ventral part of the striatum is going to be involved, and it's going to encode information about how terrible this fox odor is, and somehow that information has to get up into the dorsal striatum to help inform the dorsal striatum about what behavioral sequence is to be expressed next. That is a major problem and an important one in my lab, and if you want to come to my lab and work on that problem, you give me a call, because it's super important, it's really interesting, and it's something that we're thinking very deeply about.
Well, I'll let Christina know. And then there's another student of ours, Hector. He says, based on the data from the experiment where the lesion animal did not respond to the TMT, do you think that the dorsal striatum could play a role in approach avoidance conflict?
Yeah, that's another wonderful question. So the striatum has been, I would say, underfractionated. In other words, it's probably true that different parts of the striatum play different roles in mediating different kinds of behavior. And so people are beginning to kind of, with greater anatomical detail, try to understand which behaviors are controlled by which parts of the striatum. So my friend and collaborator, Bernardo Sabatini, has recently identified a part of the striatum that seems to be involved in licking, for example, licking behaviors. My friend and collaborator, Nao Uchida, has been working on the tail of the striatum, which seems to be involved in similar kinds of approach and avoidance behaviors to the ones that are being referred to here. And so I think in the end, my suspicion is that most behaviorally relevant information has to impact the dorsolateral striatum, because we do think that the dorsolateral striatum is actually building these moment-to-moment behavioral sequences. But it's probably true that other parts of the striatum are also kind of participating in that process and maybe, you know, encoding key information about approach and avoidance type conflicts.
Then there's another question. It says, did you see any sex differences in this?
Oh, that's such a great question. My former, there's a technician in the lab who was a co-op student for us. He's a technician in the lab named Ralph Peterson. And Ralph now has graduated and is now a graduate student at NYU. I'm super proud of him. And Ralph has been working on this problem. And so we're in the middle of writing a paper about these sex differences. And Ralph's super excited about it. And the short answer is, if you look in the open field and you compare males and females, we see very subtle differences between males and females. Okay, so it's extremely subtle. We've also done a very careful study of female behavior across the estrous cycle. So I'm sure you're aware, you know, there's a big, I wouldn't say controversy, but there's a big push at the NIH to make sure that everyone includes female and male subjects in all of their research. And some of the pushback against that has been, oh, well, you know, it's really, really hard because you have to measure the estrous cycle, where in the estrous cycle a female is, because her behavior is going to be completely different depending on that. And there's really no evidence to support that point of view, none, right? But people say it anyway, and it gives them an excuse not to do the right experiments. So we went ahead and did the experiment. We asked, you know, so is behavior really different across the estrous cycle? And the answer is no, it's entirely identical. We have the best lens on behavior, right? I mean, we have like this incredibly powerful lens. We couldn't discover any syllables or sequences that were changed in the open field across the estrous cycle. And that tells you that like that's not actually an important source of variance. And so we're excited to get this paper out. I'm really proud of Ralph for working on it. I think it's going to be an important contribution, and it just emphasizes how important it is to always include males and females in all of the experiments that you do.
Yeah, that is really, really influential. Thank you for visioning too. So we have a final question because we want to have Tari share with us the great stuff that is available at Harvard. There's one last question for you, Dr. Datta. It says that mixing mice with neuronal damage and non-damage, do their behavior change since they are interacting with healthy mouse?
Oh, that's a really great question. That experiment we have not yet done. So that's a good experiment and one that we should think about doing. If we take mice that are of mixed genotypes, like wild types and mutants, we take mice in which we're manipulating the brain activity in one mouse versus a normal mouse on the other hand, what do we see? So those are experiments we're working towards. And I'd be happy to come back in a year and tell you how those experiments go.
Great. Okay.
Carmen, let me ask a question, please.
Of course.
Mine is more like technical.
Mm-hmm.
We teach students how many, how hundreds and thousands of neurons there are in the brain.
Mm-hmm.
And then you put your microscope and you see behaviors that you can define by a few hundred neurons that are.
Yep. That's right.
Can you talk about this? And,
Yeah, that is a deep question, actually. So, and it gets to something that's very important that's going on in the field of neurobiology broadly. So, it's only been recent that we've been able to build lenses that let us characterize behavior, not just MoSeq, but other kinds of lenses too. Like the tools for understanding behavior, they're undergoing a revolution, basically. And so the way we quantify and think about behavior is really changing. At the same time, the tools that we have for measuring neural activity are changing. So it used to be that it'd be a huge miracle to record a single neuron in vivo while the animal's behaving. Now, in my laboratory and other laboratories, it's in a single experiment to do hundreds is typical. It's not abnormal, right? We have these cameras, we have these very dense electrical probes. And so we're learning a lot more. We have access to much more information about the patterns of neural firing in all sorts of different regions of the brain. And at the same time, we have access to information about the ongoing behavior of animals. And what we're learning from these experiments in general is that almost wherever you put your electrode or wherever you put your microscope in the brain, everywhere in the brain seems to have some information about an animal's ongoing behavior. It's like the whole brain is built to support an animal's naturalistic pattern of movement. It's basically the thing that the ethologists would have predicted 50 years ago, that really the brain evolved to support movements and allow animals to explore. And we're really getting the sense that behavior is much more strongly represented than you ever would have predicted otherwise. So the challenge going forward, as someone who's interested in this as a problem, is to figure out why certain types of behavior are represented in certain parts of the brain, and that's the thing we're going to sort out. Right? That's the future, because behavior isn't uniformly represented everywhere in the brain. There are differences in different brain regions. But what's been surprising is exactly what you're saying, which is that I can look at 200 neurons in striatum and predict what a mouse is going to do every 300 milliseconds. That's shocking, right? And it tells you, I think, something about the importance of ongoing behavior to the brain's underlying function. And I think I'll leave it at that, but I think that's my sense of it.
Okay. Well, we want to thank you.
I just have a quick question. Very quick question. I just wanted to know if the data regarding dominant and subordinate mice, is it available? Is it published?
It is not. It is not published. It is therefore not available. We're working on it now. We've only recently achieved this goal of being able to do multiple mice together, and I'm happy to report our preliminary results. They suggest things are going to be very promising, but it's going to take us a couple of years probably to really work that out, to be honest, to do a good job. But it does seem very promising, and we're looking forward to putting a story together.
Okay, thank you.
Well, maybe Viviana will apply for it. She sounds very interested.
Excellent.
She's great. So now, let's see. Is any other questions so we can go toward Daddy? Any other questions for Dr. Datta? Thank you, Dr. Datta.
Thank you so much. I appreciate it.
Sure, thank you. And so, Daddy, take it away.
All right. Well, first off, I just want to say how impressed I was by all of your questions. I work primarily with our graduate students, and I have to say, you could teach our graduate students a thing or two about how to ask good questions after school. So I am seriously impressed. So kudos to all of you.
We try our best.
Yeah, that was fantastic. So yeah, so just to introduce myself briefly, as Carmen and Bob mentioned, I work for the PhD program in neuroscience at Harvard University, at Harvard Medical School. I did my PhD with Bob, so I totally understand the allure of the research in that lab and why you get excited about it. It's very cool stuff. And just 1 minute of bio before that. Before starting grad school, I grew up in Salem, Oregon, so West Coast originally. I went to Oregon State University for my undergraduate. I then worked as a research assistant in a lab for a couple years after undergraduate. I worked in a lab in Boston, and then at that point I decided to apply to PhD programs. I earned my PhD at Harvard with Bob, and after that I chose to pursue educational career because I am just really passionate about education and training of the next generation of neuroscientists and creating our programs that are, you know, as inclusive and open and accessible to all of the fantastic scholars that I know are out there. So I derive great joy from being able to share all the opportunities that we have in our community and elsewhere as well. So just put that out there that I hope that many of you will contact me after the presentation today, and we can chat and, you know, about everything that Harvard has to offer, but also just in general, I'd love to hear about some of your research and your stories and get to know you a little bit better. So please do reach out. A quick disclaimer in that I was told that these sessions have been going until 6:30. I actually teach a class that technically starts at 6, and I pushed it back to 6:15, but I am going to have to run at 6:15 tonight. So I just want to put that out there that that's certainly not you know, any indication of me not wanting to be here. I wish I could hang out all evening with you, but I have put in the chat my contact information so that you can email me, and I also just put into the chat a link to a Google Doc. This is just a couple different slides, and rather than you know, presenting it and going through it right now, I figured that I can talk a little bit about some of the highlights of some of the offerings at Harvard, but you can look through those few slides that I just put in the chat on your own time as well. I'll also mention the first slide in that little document outlines our graduate program, PIN, the neuroscience program. We're going to be doing a number of different events over the coming weeks and months at conferences. We're going to be hosting our own virtual open houses that anyone can register for. And the timing of the event tonight is also especially convenient because next week is the BP Endure grad fair, and myself and some of my colleagues and students in the program will also be there as well. So since we have limited time tonight, I do encourage you to stop by at that session and any of them. And one other thing I'll note is that in that document, there's a place where you can submit your email to me to connect and be kind of added to an email list that I'll be sending out to remind students of the various recruitment events that we'll be doing. And also there's just a brief survey in there to collect information from you if you're interested in undergraduate summer research programs, post-bac programs, the PhD program, other things at Harvard, just so that I can answer your questions directly or connect you to some of my excellent colleagues across the university who could answer the questions even better than I could. So with that aside, I just want to broadly introduce, you know, what are some of the opportunities to do research at Harvard. So all of you as BP Endure scholars, I know are getting fantastic undergraduate research experiences right now. So many of you may be thinking of going straight to a PhD soon or as soon as you graduate. So in the PhD landscape, Harvard actually has a number of biological science programs, 14 programs under this umbrella called Harvard Integrated Life Sciences, or HILS. You'll see this acronym in the slides that I shared. What that means is under this big umbrella, you can rotate in laboratories or join thesis laboratories in any of these different programs, no matter which program you actually apply to and get into. So it's basically we have this big structure at Harvard that gives you lots of opportunities to do research in the types of labs that you want. So for example, PIN, the Neuroscience PhD program, we are one of these 14 programs, and certainly students apply to our program, and I'd love to see many of you apply to our program. But students also find that maybe they want to instead get a curriculum that's not so focused on neuroscience, but maybe more in the biological, biomedical sciences, a broader base where they're taking biochemistry and genetics, whereas our curriculum is very neuroscience focused. And just to say that this means you can apply to that other program, biological, biomedical sciences, and you can still do your neuroscience thesis work in a neuroscience lab, for example. So there's a lot of integration there. So I just encourage you, when you're looking at PhD programs at Harvard, I obviously can speak most directly to PIN, which is the program I work with, but there are many other options at Harvard as well, including if you want to do neuroscience research. There are other ways to do that as well. So I just want to give kind of a brief overview of PIN, our neuroscience program, and for those of you who are following along on the Google Doc, this would be the second slide there. First and foremost, I want to say that PIN, as many PhD programs in the country, completely covers all your cost of tuition. So you don't have to pay tuition to go to graduate school. And we also pay you an annual stipend to do research because we recognize that's going to be a full-time job. Your training, your studies, we don't want you to have to worry about other things outside of that. The annual stipend for PIN as of this year is just over $40,000 a year. Boston is an expensive city to live in, but many of our students do end up rooming together. We have a really good network, myself and the other members of the leadership team for our PhD program to help students connect and find roommates and things like that. So students tend to be okay with that. Our program also offers, for those of you who would be moving to Boston, a $1,000 extra moving supplement to help offset the cost of that. But just to say up front, we often get questions about funding for graduate school. So your funding would be completely guaranteed through our program for as long as it takes for you to do your PhD. So you don't need to worry about that. Students who might be interested in applying to separate fellowship programs like the NSF fellowship, or if you have other ways to secure funding, that's awesome, but it's certainly not necessary either for admissions to our graduate program or for once you're in our graduate program. It's kind of an extra bonus for your CV. Our PhD program has about, and if there are any questions, yeah, I guess you can put them in the chat or you can also interrupt me. I said I'm trying to give a quick overview, but certainly if people have burning questions, I can answer those too. So just quickly, our graduate program has about 20 students admitted each year. So at any given time, our program as a whole has about 120 students in it. We have a good mix of international students, domestic students, students from different backgrounds. I would say the PIN community itself, when I was interviewing for graduate programs, one of the primary reasons I chose to come to PIN was because I found the student community to be so engaging and welcoming and supportive. In particular, what really impressed me when I was interviewing at the program was that the students all knew each other, not just those in their own class, but students who were sixth-year students in the program knew the second-year students in the program. And so that was really evidence to me that they were all supporting each other. So I would say we have a really strong student community. That's one of our best assets. Our students also within the community, not only just broadly finding support through student-run events, but there are also a number of student affinity groups within PIN, as well as the broader Harvard community, that help people find colleagues and peers from particular affinity groups with which they identify. So there's a underrepresented scholars in neuroscience group that's actually run by PIN students within our PhD program. And they do fantastic things like practice. They do social events. There's actually an event tomorrow night. Social events as well as professional development events in terms of giving a space for people to give practice talks if they're giving a seminar or if they're preparing for a committee meeting where they have to give an update. And so that's a really good group. We also have a Women in Neuroscience group that's largely housed in our department at the medical school. More broadly, there's a biomedical scientists, minority biomedical scientists of Harvard group, MBSH, that expands not just, encompasses not just PIN, but other PhD programs as well. And there are many other affinity groups. I linked a number of them in that Google Doc, but there's a student organization for Puerto Rican students, Latinx students, LGBTQ students, so across the broader Harvard landscape. I'd also say our graduate school, writ large, has a fantastic team of women who run their Office of Diversity and Minority Affairs and oversee all of these different student affinity groups, and they are just amazing allies. So again, if you email me afterwards and want to be put in contact with any of them, Dean Sheila Thomas or Associate Director Karina Gonzalez Herrera, they are both fantastic, and I work very closely with them. So that's kind of like an overview of the PIN student community in terms of what your training would actually look like in our program. The first year, our students take a short MATLAB bootcamp course that teaches you a little bit of coding in the MATLAB programming language, as well as some statistics. This takes place the summer right before you start school, so for 2 weeks in August. There also is a year-long course that all of our students take called the Discipline of Neuroscience that's intended to give all of our students, regardless of your particular areas of interest or your backgrounds, kind of a common understanding of the lay of the land of neuroscience. We do get applicants who have studied lots of different backgrounds. I, as an undergrad, studied biochemistry and biophysics. We get people who from psychology, from math and physics, from biochemistry, neuroscience. So that year-long curriculum is a way to kind of bring everyone up to speed. And then beyond that, our course curriculum consists of elective courses so that students have the option to individualize their PhD training based on your specific interests and what you want to pursue. So that's kind of our coursework. And then in our PhD program, students do try out labs their first year. They do laboratory rotations, which we're very flexible about in terms of how many you do, how long they are, when you do them. You work with advisors within the program to help you figure that out. And then by the end of typically your first year, by fall of your second year, you've found a home to do your thesis research. So that's kind of the general structure. I see there are a couple of questions here, so I just want to try to answer those. And again, if you have any more questions, put them in the chat here. And because I'm going to have to run in a couple minutes, I don't know if Bob is able to stay on a few more minutes. He can also, he knows some stuff about Harvard a little bit.
I'd be happy to stick around.
But anyway, so the first one question, the living expenses are paid by the students with their stipend. Yes, so the stipend goes towards essentially cost of living. And as I mentioned, as long as in most cases, you're not going to be able to have like your own apartment in downtown Boston on that stipend. But for students who live with a couple of roommates or classmates from the program, I would say, having gone through that myself as well, it certainly is a comfortable, the stipend is allows you to live comfortably, covering the living expenses in Boston, as well as even being able to take advantage of some of the great things in the city. We have sporting events, concerts, history stuff, lots of great art in the city, and there are fantastic student discounts. So that's a really useful thing too that makes those more accessible. We do have an MD PhD program, and IMSD, is that a, Carmen, is that a summer research program, IMSD, Viviana?
It's for, I think it's maximizing student diversity in graduate studies. It's funded by the NIH.
Okay, so.
My understanding is that we do actually have an IMSD program.
Okay, through Sheila's office then, Bob?
Sorry?
Through Sheila's office?
Yes, yes, exactly.
Okay, so I can look into more details about that, Viviana. There is an MD PhD program. To apply for the MD PhD, you actually apply through the medical school, not through the PhD program. So the way they do that would be on the medical school application cycle, which is slightly different from the PhD cycle. I will mention that our PhD, if anyone's thinking of applying this year, both our program as well as most, if not all, of the PhD programs at Harvard have a similar application deadline of December 1st. So that's why in that slide deck that I gave you, that's why over the next couple months we're holding lots of sessions. We're going to be talking with lots of students. The 2 virtual open houses that we're planning to host ourselves One of them will be on meeting people in person and breaking down the admissions cycle, so the admissions process, so you understand that a little bit more. And then the one that we'll have in November is more of an open Q&A, as maybe you've been preparing your application and you have some extra questions, as well as a student panel, so you can meet some more people. And then one last question I see here: how many graduate students do you take on each year? So we end up having an incoming class of about 20 students. So in the grand scheme of programs, it's large by some aspects, certainly, but compared to some of the other programs at Harvard, like that BBS biomedical science program that I mentioned earlier, they have an incoming class of 60 to 80 students. So by those standards, we're small. So we're kind of a medium-sized program. But so that's my spiel. Oh, great question. Thank you about the GRE. GRE is not required, and in fact, we will not look at your scores if you submit them. So do not submit GRE scores. I should add that to my my snapshot. Thank you so much.
Okay. Well, for everyone that want to contact Daddy or Dr. Datta, do you have their email? Please do so. And they're more than willing to answer your questions. And of course, our BP and DUR students will have an opportunity to see Daddy again next week. And obviously at the open house and activities that they have.
Yep.
And so thank you, Daddy, for being here.
Yeah,
Dr.
thank you.
It was a pleasure.
Thank you so much. I really had a great time.
Yes, I really appreciate this. And join us next time, because this goes on until next Wednesday, when we will have Dr. Ardreen Boyer, who is a physician scientist at the Sloan Kettering's Cancer Center. Is going to talk about a very interesting topic about how cancer affects the brain. So that's another approach that the students will have the opportunity to listen to, about great research on that aspect. So thank you so much for being here, and have a great evening, everyone.
You too. Take care, everyone.
Hope to see many of you again. Thanks.
Take care. Bye. Thank you.